It's my pleasure to introduce Ashley Zehnder. And so I'm actually really excited to hear this talk and learn more about Earth's most resilient creatures. And so Ashley is a co-founder and CEO of Fauna. It's a company that was launched from the Age 1 Accelerator in 2018 for companies focused on aging research. And so Ashley's been recognized for her work by the SF Business Times Women Who Lead in Life Sciences, an emerging woman founder in bio by the WAVE Summit, as well as representing Fauna Bio for the Fortune AI Minute. And she is a boarded avian veterinarian. And her previous research focused on translational science, earning a PhD in cancer biology from Stanford. And her research work spans over 20 peer-reviewed publications, including in Cell, Nature, and Nature Medicine. So please welcome Ashley.
Hi, good afternoon. If I'd known you were going to read the whole bio, I would have sent a shorter one. Okay, so I'm Ashley Zehnder. I'm the CEO and one of the 3 co-founders of Fauna Bio. What we do at Fauna is take advantage of, in some cases, hundreds of millions of years of adaptive evolution to find better therapies to help humans live longer and healthier lives. And we can do that because what people are often unaware of is that these animals are doing this using the same genes that you and I have, but in very spectacular ways. And now the science has come along enough that we can start to use these insights to derive new and novel therapies for a number of different diseases. So typically, oftentimes how we do drug discovery in human biotech today is that we think about looking at genes and gene expression in humans that are either sick or in some cases getting sicker. So looking at genetic mutations that are associated with humans that are predisposed to certain illnesses, or looking at how genes change when humans get a disease or are progressing along that disease pathway. What is sometimes more helpful is to look at humans that are naturally resistant to certain types of diseases, and there are biotechs that are doing that as well. One great example of that is humans that naturally have mutations in a gene called PCSK9. Humans with these mutations often have a naturally lower cholesterol level that led to the development of very successful PCSK9 inhibitors. The problem is that there are just simply not people that are resistant to all the diseases that plague us today as a species. Fortunately for us, many animals do, and that's really the basis of what Fauna Bio does. So I will spend some time today talking about one amazing creature called the 13-lined ground squirrel. This is a prototypical hibernator, deep-bodied hibernator, but they are prototypical of a number of different species that have a very similar biological pattern that end up being resistant to a number of different diseases. What's remarkable about these guys is that they hibernate for 6 to 7 months out of the year. They take their body temperature down to almost freezing. They stay there for a couple weeks at a time. While they're down there, they're almost 90% hypoxic. Their neurons in their brain physically retract. They're clinically brain dead. They're flat EEG. They become hyper-tau phosphorylated, very similar to what happens in an Alzheimer's patient. If that wasn't cool enough, what happens is after 2 weeks, they rapidly rewarm back up to normal body temperature in just about an hour. And so all of those changes have to reverse. It's very similar physiologically to what happens during a heart attack or a stroke. So you can imagine a human being surviving what ends up being the equivalent of 25 heart attacks over a 6-month period. I don't know that many people in the audience that could do that, but these animals do it every year, right? And they come out with normal-looking hearts. And I'll show a little bit of an example of that in a moment. When they're doing these rapid rewarming periods, they're increasing their metabolism 235-fold in an hour. My CSO, Dr. Katie Gerbeck, likens it to going from a dead sleep to biking the Tour de France in an hour, right? When you're looking and working with large pharmaceutical companies, which we do and I'll talk about briefly in a moment, these companies are looking for ways to counteract some of the effects we're seeing with some of the very successful obesity drugs that are on the market today, namely that people tend to then lower their own metabolic rates. You have to stay on these drugs essentially for your lifetime or you'll start to rebound body weight because you've lowered metabolic rate. These animals can increase their metabolism massively using core mammalian genes. The cone photoreceptors in the back of the eye physically melt every couple of weeks when they go into hibernation. You watch them on EM, they just fall apart. It's amazing, and they regrow, and they fall apart, and they regrow. So we're looking at mechanisms of retinal repair and healing. Their GI tract, they don't eat for 6 months out of the year. Their GI tract has to maintain stability and integrity during that entire period of time. They're also not moving during this whole period of time, so talking about muscle wasting mitigation, right? They're just laying motionless under the ground for 7 months. And they have to endure several cycles of low oxygen and ischemia reperfusion injury throughout the entire cardiopulmonary system. So this is just a sampling of how amazing these animals are, and they're not the only ones that can do this stuff. But just for interest, this is what one of these things looks like before they go into hibernation, right there. So I thought it would be fun, because it's right after lunch and people tend to fall asleep, that I thought it would be fun to try and play some videos of what this looks like. So this is a thermal cam image from one of our animals up. We have a research facility in Oshkosh, Wisconsin, of going for an animal that's going, you can hold them in their hand, they're physically cold, from about 10 degrees Celsius, warming up very rapidly, in this case up to about 25, but they'll get all the way up to 37, and this happens in a very, very quick period of time. It's very cool. Makes really good videos too. So this is an example of what kind of comparing human biology with, we'll show squirrel physiology in a moment. So this is normal human cardiac histology up in the top. This is what a human heart looks like 1 to 3 days after a heart attack. You get this disorganization of muscle fibers. You could be staining these tissues for collagen. You have collagen fibers going up. And what happens in humans is that we often go on to form scar tissue, right? We end up with scarred hearts that ends up leading down the road, oftentimes to heart failure. What happens in these ground squirrels, they start with normal hearts, and if you damage their hearts in the summer, interestingly, they're just like any other mammal. But in the winter, they're protected. During these rapid rewarming periods, very similar kinds of damage to what happens in humans. Their hearts are disorganized. If you stain them for collagen networks are going up. But, interestingly, this is the cool part, they are able to repair that damage and go on at the end of that season to have normal-looking hearts. It's crazy, and people doing this like, yes, it's real. What's even cooler is that they're not unique within mammalian species. This is a trait that we see replicated across the mammalian tree of life. This is data that was gleaned from our collaboration with the Zoonomia group that is operated out of the Broad. And what's really interesting, too, is if you look at overall lifespan in many of these species that have this trait of hibernation, they live significantly longer than species that are similarly sized. I gave a talk at the summit last year that really went through more of the links between hibernation and aging biology. So there's a lot of research going on looking at those links. This is everything from these animals can more highly protect their DNA, they can protect and elongate their telomeres, they can obviously have enhanced mechanisms of cellular regeneration and repair. They also have ways to increase their mitochondrial function and reduce oxidative stress. So there's many different layers about how these animals repair and regenerate tissues. What I'm going to try to talk about more today in this talk this year is how we as a company translate this really extreme and highly adapted biology into benefits for people. Because I mean, as much as I love ground squirrels, they don't really need my help, right? So to do this, we built Convergence, which is our platform, our aptly named platform, that functions to merge human disease data with animal resistance profiles and then look at the enrichment between the two. So part of what drives the work that we do are these highly proprietary data sets we generate from animals that nobody else in the world has access to, and then aligning that with many, many different kinds of human data, and I'll show more about that in a moment. That enables us to build not only our own pipeline, but also allows us to work with pharmaceutical companies who are looking for solutions to really hard health problems. And this is typically the pattern that I've been seeing in the companies we're talking to are companies that have had some level of success in really hard disease areas, obesity, Alzheimer's disease, heart failure, and now they've been told, hey, you need to build a pipeline in these hard disease areas, and they've sort of tapped out where they normally look. They've looked at all the human data, they've looked at all the mouse and rat data, and they don't really know where else to look. We give them a place to look for them. Because we're working with really extreme physiology, we don't need a lot of samples, actually. So our primary pipeline programs, which I'll talk about briefly, we found with 60 samples from the ground squirrel, right? And we found enough signal from that small number of samples to find really powerful and differentiated biology. This allows us to do pipeline development quite capital efficiently, which allows us to maintain a diverse pipeline and also be able to do work for pharma partners more efficiently. We're looking at genes and pathways that are highly conserved across many hundreds of mammals. This is important because it means that these are pathways that are more likely to be functional in disease and are more likely to translate into humans, and we'll talk a little bit more about that. So that was really the major finding from this group of papers that came out in Science at the end of April. It wasn't an article, it was the whole issue of Science. So if you haven't looked at this, it's a really cool set of papers. Eleanor Carlson was one of the co-authors on these papers. She's a SAB member for Fauna. And really the idea behind Zoonomia was looking at getting enough data from animals to be able to look at the human genome at a single base pair level and say how conserved are each of those base pairs within the human genome. And you need this many species to be able to do that. Why that matters is that if genes haven't changed in over hundreds of millions of years, it likely means they're doing something important, right? And they're more likely statistically to contribute to disease. And so now we have the power to look at these, and now we can link these genes to human disease faster than other people because of the data sets that we use. So how exactly do we do this, right? We start with the data protection signatures, and I'll show an example of how we do that in a moment. We look at tissues and time points where we know animals are protected from damage. We align that to human disease states, and then we look for enrichment. We test all of our predictions with our own labs up in Emeryville, California. So we do our in vitro work in human cells and human cell lines. We also, as I mentioned, have a facility in Oshkosh, Wisconsin, where we can do our own work there. But everything has to pass that sort of, does it function in a human cell model filter before we move forward? And then we integrate with a number of other different data sets, what I'll talk about in a minute. And then really critically, we map gene expression signatures to existing compounds, so we can say, okay, this is a gene expression signature that looks protective. Can we find a compound that mimics that protection? So I'll show a real quick example of how this works. I did this a minute ago. There we go. So this is a little video snippet from one of our modules on Convergence called ORCA. What ORCA does is it aligns hibernator protective signatures to human disease states. And so in this case, this is data from our heart networks. What this looks like. I'll pause that for a second. These are genes in blue that are down-regulated at time points where hibernators are protected from damage. They are ringed in yellow if they're also up in humans that have heart failure. And so then we can look not only at a specific gene, but also an entire network that is then statistically enriched for this opposite protective biology. And then, as I mentioned, right, we validate all of our programs first in human cell lines. This is a study looking at knocking down a gene that was down in hibernators at protective time points, showing that in vitro we can double human cardiomyocyte survival in the context of oxygen and glucose deprivation. We can then take those same genes and knock them down in rodent models of cardiac damage, in this case a coronary ligation study, where we validated not only this gene, but then 3 independent genes from different networks and 2 compounds that we found through the same approach. And how we map these to compounds is we take these gene expression signatures that are enriched and validated in our plot process. We then map those to chemical library signatures, and we can identify small molecules that will mimic this protective signature. What we've been doing more and more of in the last, I would say, 12 months or so is really looking at better ways to integrate and learn on the data that we've collected. And it's really coming out of a process of getting enough data and also enough complexity of data to be able to do this more advanced type of analysis. So here, this is just a screenshot of some of the data types that are in Centaur, which is our knowledge graph, which is now cataloging more than 18 million relationships between the animal resistance data, but then all of these clinical and human genomics resources. What that allows us to do now is start to use much more cutting-edge graph neural network techniques to be able to traverse this knowledge graph and be able to make new predictions between the role of genes and particular human diseases. What we found really interesting is when we ran this model in 2 different ways. One of them was with no animal resistance data, where we tried to say, can we just show a link between a gene and a human disease? We got an ROC curve that was, kind of in the high 80s, which is like okay, but it's not stellar. A lot of people would much prefer to see ROCs that are over 0.9 if we're going to be using them for anything meaningful. But if we just add in this, what looks like a small amount of data, it's actually not a small amount of data. These other data sets happen to be a little bit bigger. If you add in just the animal resistance data, right, you can massively increase the predictive power of this to now be over 0.9. And we're continuing to refine this as we go. In my head, because I'm not a computational biologist, I think of this as putting a key into a lock, right? You have all of this complex human clinical genomics data. How do you get the best insights out of it? Sometimes it's just a little bit of the right data to do that. And so by doing this, then we can get insights into multiple different disease areas. And if you survey, where are the top pharma companies in the world looking for innovation, the vast majority of them are relying on human genomics to build drug programs and also build programs around precision medicine. This approach can really help all of those companies do that better. And so now we're generating a significant amount of revenue in this year alone by doing partnerships with big pharma. So how do we actually build our own pipeline of assets using data from extreme genomics? First of all, I have an awesome team. So again, I'm not a hibernation biologist. I'm not a computational biologist. I am not an experienced drug developer, right? I just work with them. So my background is in veterinary medicine. So my expertise is really at that interface of human and animal physiology. I'm more of a clinical translational person. My CTO, Dr. Linda Goodman, has a background in human genetics. Frankly, got really frustrated with how hard it was to find impactful human drug targets by using human data alone, and turned to the process of comparative genomics to be able to do that more efficiently. My CSO, Dr. Katie Graybeck, also has a PhD in human genetics, spent that entire PhD studying the biology and the genomics of the 13-lined ground squirrel, and built some of the initial biobanks that we have now, and is leading our entire research effort to find and validate targets from that and other species. Brian Berkey joined our leadership team about 2.5 years ago, having spent a little over 20 years at Novartis, building drug development programs really from the ground up, particularly in cardiometabolic, and then working on some smaller startups before he joined us at Fauna. And then bringing on board folks who have expertise not only in drug development, but medicinal chemistry, clinical development, comparative genomics, and then thinking about corporate strategy and partnering. So, by the numbers, right, what we have done since starting Fauna a little over 5 years ago is we opened up a pipeline that can address very large markets, right? These are diseases with high unmet need, with a number of patients who could benefit from these assets. We've actually been able to build real drugs out of this approach, right? It's not a theoretical any longer. We have novel chemistry against known and validated targets in new disease areas with high unmet need. We've been able to generate revenue already from partnered programs. And we've done this quite capital efficiency. Or when you survey platform companies, particularly genomics platforms, oftentimes they'll spend sometimes 10x this amount of capital to build a pipeline that is as far along as this one is. And I showed data from the cardiac assets that we have, but we've also been able to validate compounds and genes in pulmonary disease, in obesity, and within CNS disorders as well. So this is a current snapshot of where the pipeline is today. So our lead program, FON1003, is a small molecule that we found based on mapping small molecules to gene expression signatures of ischemia reperfusion injury and also hypoxia during hibernation. And so this is a molecule against a well-known human druggable target that now we have evidence for it in a new disease area and where Fauna has derived novel and proprietary chemistry. We also have a second program, FON264G. It really shows the modality agnostic ability of our platform, where we can find specific genetic targets that we can then apply to certain diseases. So in this case, this is a gene that we found by looking at neurons and how they're protected during hibernation. We can now apply this to a disease like retinitis pigmentosa, which has patients with multiple different mutational backgrounds. And then we're working on partnering activities across many different disease areas, and I'll be able to talk more about that in a couple of weeks. And this is just a little bit more detail about some of our primary pipeline assets here. The small molecule I just mentioned, again, much safer mechanism of action for patients with subsets of ILD, pulmonary hypertension, and potentially some patient populations within HFpEF as well. Interestingly, when we look at our mouse and rat studies where we treat animals with FON1003, we've noticed this trend that we're actually decreasing several different types of age-related pathways within these animals, everything from senescence markers to immune function and inflammation. Similarly with FON264G, this is a gene that's highly related to metabolism. So this is a gene that people have looked at in the context of cell senescence, as well as hematopoietic stem cell function. Interestingly, this is a gene that's really required for ketone utilization, so cells will upregulate this gene to utilize ketones more effectively. And then this is a pathway that we're looking at, not only for retinitis pigmentosa, but also for potential neurodegeneration. So, at the end of the day, right, we have data and assets from one species and really one disease area that we've already built a significant amount of value. But as I mentioned at the beginning of this talk, right, this is a strategy that we can replicate across many different species around the evolutionary tree. And so this is really just the beginning. I wanted to highlight, as we're rounding out the end, I'm almost done, our first non-hibernating collaboration. So hibernators are amazing, but they're not the only amazing species out there. There's a group of species that are the spiny mice, and there's a group of researchers in Florida doing work with these guys. They can repair multiple tissues in the body without scarring or fibrosis. So I wanted to show 2 quick videos. This is not our work. This is work that was published last year in DevCell. This is get this guy over there. Here you go. This is a normal mouse. 8 weeks after a spinal cord transection, right, still fully paraplegic. This is a spiny mouse. It's 8 weeks after a spinal cord transection, right? not paraplegic. And if you look at these under histology, these animals have regrown their spinal cord without scarring. And so they can do this not only with the spinal cord, but also with parts of the brain, the kidney, and within the skin. And so we're starting to look now at brain and skin sections from spiny mice that we're just starting to work with now. So with that, I will take any questions. Thank you.
Great. I'm a big fan of the African spiny mouse, actually. So, interesting thing about these, though, is that it's not just like a single gene target. And so it ends up being this multifaceted aspect where a lot of the genes that have come out in the proteomics hits tend to be things that also promote cancer, right? So how do you address that in this issue where it's like, you can regenerate these things, but you're also going to dramatically improve cancer because this is a mouse that lives for 2 or 3 years.
Sure, exactly. And I think what we like to do here is not over-index on traits in any one particular species, right? Part of what we like to do is take data from a species, but then really look at how it's changed and aligned with other species that maybe have similar protective capabilities, and also with human disease, right? So we see enrichment in a pathway with regeneration in spiny mouse, but, oh shit, it also gives people cancer, then we will probably look at something else, right? So what's interesting about some of the hibernation models is that a lot of the genes that they're turning on can be sort of dangerous in the long term. The heart reparative gene pathways are the same ones you see in hypertrophic cardiomyopathy. What's interesting about those species is how they turn them off. So a lot of it is very time point specific, right? You want to catch these gene expression pathways in the right context. Are they turning on or are they turning off? And how are they reparative, and when do you need to sort of dampen them back down? And so that's a lot of what we work on with the hibernation models is when is the damping off point? And that it's just really critical to get time points and the right data at the right time point. So I totally agree with you, and that's why, like I said, we never try to look at one species kind of in isolation. We always kind of look at it in the context of other species that are similarly protected, and also, like I said, 80% of the data on our platform actually ends up being human data for that reason. Yeah, it's a great question. Nothing is as simple as it appears. There's one. Oh, there's two here, one here, and one there. Yeah. Where? I don't know. I think they're in there.
Ashley.
Yes.
It's absolutely amazing progress. I'm super glad to see this. I'm just wondering. So, playing the devil's advocate here, evolution tends to change many things at once, and something as complex as regeneration probably takes multiple changes. So, but as a drug developer, you only have like one drug, one hit usually. So how do you think about like the translatability of these things?
What we've seen, I'll try to be short and I can grab your question as well. What we've seen is that oftentimes we're hitting an entire network of genes, very often, kind of like what you say. And what we end up testing is we end up testing the hubs of these networks to look for phenotypes that are then related downstream to multiple other pathways. And so we found that within the cardiac data, there was a hub of a gene that was 30 genes that were changed similarly. And then we could start to trace that down and figure out sort of what's the right druggable node, right? So it's never a single gene. It's always a set of genes. And so we can either look specifically at those genetic networks, or if it is a signature of, say, 20 to 30 genes that may not even be in the same network, that's where you start to map them to chemistry and say, okay, where do we see drugs that exist now that are hitting a variety of these things? And you start to optimize that chemistry for a disease indication. So we can sort of do it both ways. But I agree, it's very rarely like a single hit, although in the cardiac work that we did, it was modifying a single gene that was protective in those cases. So it depends a little bit on the disease area. Some are more complicated than others, but it's a good question. Yeah.
Cool, thank you.
Yeah, I had a question about your Fauna 264, that ketone body. So groups at UC Davis and also here at the Buck have shown that ketogenic diet extends longevity. I'm just wondering how that might fit in the pathway, or do you have can you tell us a little bit more about it?
Yeah, that's a really interesting gene. I was trying to stay under time, so they didn't chase me off the stage, but it's a really interesting gene because the hibernator brain, as I mentioned, sort of goes flatline during hibernation, except for 2 cell populations that they have to stay functioning during hibernation because they have to wake up. It's a little bit like the pilot light in your stove or the part of your computer that wakes it back up when you close the lid. The gene that those cells turn on in your brain is this gene 264. And what it's doing is allowing the neurons in your brain to stop using glucose, which is not available because they're not any for 6 months, allows them to use either fatty acids or ketones, which are available. And so that's what this gene does. It allows multiple different cells to be able to switch from using glucose to using beta-hydroxybutyrate or lactate. And that becomes particularly important for retinitis pigmentosa because there's this odd interplay between the RPE and the cone photoreceptors where the RPE starts soaking up all the glucose, the cone photoreceptors are starved, but if you can allow them to use lactate, it's great. That's not the only place where it's useful. People have looked at ketogenic diets as being helpful for diseases like ALS and improving mortality in patients with ALS as well. So I think there's many other applications for that, but it's a gene that you don't want to just go willy-nilly overexpressing all over the body, and so we're trying to take a very targeted approach to that. Yeah, it's really cool. It's a really cool pathway, though. Yeah. Other questions? Oh, one more. If I have my time. Yeah. You can shout. I can probably hear you. Oh, Lord. Okay.
Space travel. One is the African lungfish can hibernate. I'm sure you know this, right? It's 10 years.
There's a lot of species that can go into this really prolonged period of dormancy, like pond turtles, right? And
Right.
Yeah.
And then the other animal is the Australian weta, W-E-T-A.
I'm not sure that
This is one of the biggest insects. The benefit is the blood antifreeze.
Yeah, for sure.
So in the human, becomes crystallized. Those are good ideas, but how do you translate this to the real life?
Yeah, we start with mammals. We get this sort of question all the time, is just how would you look at some of these even more extreme, right, species? And then you start to run into problems of translation, right? So we have intentionally maintained our focus within mammals, largely for areas of translation, right? How can we rapidly translate findings from these species to humans? And ideally, if we can find genes that are conserved across, say, a number of different mammals and, say, primates, then the likelihood they're going to translate goes up. Trying to translate sort of antifreeze molecules from some of these species is just a lot more difficult. And so we just haven't kind of gone down that road. Maybe in the future, I don't know. But mostly we try to stick within mammals, really for that reason of translation.
Talk to Elon.
I guess I should, yeah. I feel like he's probably busy right now. Anybody else? Oh, one in the back. I must have talked really fast. Okay.
It was reported relatively recently that the spiny mouse goes through menopause. Is that true, or is that like a wrong? Did you hear about this?
I've heard about that.
Okay.
I won't officially comment on that because I haven't read the paper, and so I don't want to say something wrong.
No problem. All right.
I'm not an expert in spiny mouse biology.
Okay.
We should wrap it up. Okay. Christine's chasing me off the stage. Okay.